Hello students, I am here again, Dr. Nidhi Sharma, with a new lecture video. In previous video, we studied about the cells, okay, the structure of a cell, the size of a cell, and the shape of a cell. Today we will study the two different type of cell, that is the prokaryotic cell and the eukaryotic cell. First, we will study about the prokaryotic cell. Now, as you all know, what is the prokaryotic cell? Prokaryotic cell. Prokaryotic cell. Prokaryotic cell is that cell in which there is no nuclear membrane, and no doubly membrane bounded organelles. No doubly membrane bounded organelles. Okay, so this is the characteristics of the prokaryotic cell. This is the characteristics of the prokaryotic cell. Okay, so example of prokaryotic cell is what? Bacteria and what? Blue-green algae. So this is the example of a prokaryotic cell. Now, what are the occurrence? Prokaryotic cell is placed in kingdom Monera. It is placed in the kingdom Monera. Okay, so the prokaryotic cell, it is placed in the kingdom Monera. Okay, example is bacteria, blue-green algae. Okay, or you can also say mycoplasma. Mycoplasma is also the example of prokaryotic cell. Mycoplasma. Okay, and PPLO or PPLO. Full form of PPLO I have told you in your previous lecture. Pneumonia-like organisms. Okay, so all these are the examples of the prokaryotic cell. Now bacteria. First we'll study about the bacteria. This bacteria, it is the most common type of organism which is placed among the prokaryotes. Okay, the most common type of organism which is placed among the prokaryotes. It is placed among the prokaryotes. Okay, they are generally smaller and multiply more rapidly than the eukaryotic cell. So this bacteria or the prokaryotic cell is generally what? Smaller. It is generally smaller and multiply more rapidly and multiply more rapidly than the eukaryotic cell. Okay, than the eukaryotic cell. Now, as you are seeing this diagram, are you all seeing this diagram? This diagram is a typical eukaryotic cell. Now, this typical eukaryotic cell has the nuclear membrane. It has the nuclear membrane and it is also having the doubly membrane bounded organelle. This is the mitochondria. This is what? The Golgi apparatus. This is endoplasmic reticulum. So this typical eukaryotic cell is having the nuclear membrane, that is having the mitochondria, then it is having the Golgi apparatus also, endoplasmic reticulum also. Okay, so this is a typical eukaryotic cell. As you are seeing here this diagram, this is the bacterial cell. This is what? The bacterial cell. This is the typical bacteria, which is how much? 1 to 2 micrometer in length. Okay, then this is PPLO, which is 0.1 micrometer. This is the virus, which is 0.02 to 0.2 micrometer. So these are the diagram which shows the comparison of the eukaryotic cell with the prokaryotic cell. Okay. Now the bacteria, they are found in almost every places. Bacteria is found almost in every place, like deep in the soil, sea water, or human intestine, or what the human intestine. Okay. Now we'll study about the size of the bacteria. Size of the bacteria. We'll study about the size of the bacteria. Now, what is the general size of the bacteria? Bacteria vary greatly in size. Bacteria vary greatly in size. Bacteria vary greatly in size. Okay. It is how much? It is 0.3 to 1.5 micrometer. 0.3 to 1.5 micrometer. This is the general size of the bacteria, that is from 0.3 to 1.5 micrometer. Okay. Now the shape of the bacteria. There are different shape of the bacteria. One is spherical. This spherical is called as coccus bacteria. Coccus bacteria. Okay. Now this is coccus. Then if it is rod shaped, then this is called as the bacillus bacteria. If it is rod shaped, that is called as the bacillus bacteria. Okay. Then if it is comma shaped like this, then it is called as vibrio. If it is comma shaped, then it is called as vibrio. If it is spiral, then we call this bacteria as spirillum. Then we call this bacteria as spirillum. Okay. So this was about the different shape of the bacteria. This was about the size of the bacteria, which is from 0.3 to 1.5 micrometer. Shape of the bacteria. This is coccus bacteria. This is bacillus. This is vibrio. And this is spirillum. Okay. Now we'll study about the next topic, which we will study about is the components of the bacterial cell. Components of the bacterial cell or the prokaryotic cell or the prokaryotic cell. So these are the components of the bacterial cell or the prokaryotic cell. Okay. All prokaryotic cells are similar in their organization, although they exhibit a wide variety of shapes and function. Okay. So all the prokaryotic cells are similar in the organization. Now a bacterial cell is composed of various components. Various components in the bacterial cell. Various components in the bacterial cell. There are various components in the bacterial cell. First is what? The cell envelope. First is the cell envelope. Then second one is the cytoplasm. Okay. Then the third one is the genetic material. Okay. Then we have nucleoid. Then we have the nucleoid. Then we have inclusion bodies. Okay. Then we have ribosomes. Then this ribosomes help in the protein synthesis. Okay. This ribosome helps in the protein synthesis. Now the next component is flagella. Flagella is the locomotory organ. Flagella is a locomotory organ. organ. Okay, it's a locomotory organ. Then we have pili. Then we have fimbriae. Pili helps in it helps in the sex of the bacteria. Okay, and fimbriae. So pili, it helps in sexual reproduction, you can say. Then fimbriae, it helps in connecting, helps in getting, it helps in getting the bacteria attached to the rough surface, Okay. So this was about the fimbriae. So all these are the components: cell envelope, cytoplasm, genetic material, nucleoid, inclusion bodies, ribosome, which help in protein synthesis, flagella, which is a locomotory organ, pili, which helps in sexual reproduction of bacteria, then fimbriae, which helps in getting the bacteria attached to the rough surface. So all these are the components of the bacteria. Okay. Now we will study about the cell envelope. Cell envelope. Are you seeing? This is the bacterial cell. This is a typical bacterial cell. It is a typical bacterial cell. Cell envelope. This cell envelope is the outermost covering of the protoplasm of the bacterial cell. Okay, and it is known to protect the cell from mechanical shocks and injuries. This cell envelope, it protects the bacteria from the mechanical shocks, and what the injuries. Yes, so cell envelope. Now we'll study about cell envelope. Cell envelope, what it protects the protoplasm of the bacterial cell. Okay, it protects the cell, protects the bacterial cell, from injuries. Okay, from injuries and shocks. Okay, now this cell envelope is composed of 3 layers. First is glycocalyx. This glycocalyx is also called as mucilage sheath. Okay, then second, cell wall. Okay, second is what? The cell wall. Then the third one is the plasma membrane. Okay, so these are the cell envelope. First is glycocalyx, which is called as the mucilage sheath. Then we have the cell wall, and then the third one is the plasma membrane. Okay, then now, are you seeing this diagram? You must be seeing this diagram. Okay, in this diagram, glycocalyx, this is the outermost layer and is called as the mucilage sheath. It is made up of, it's the outermost layer. It is made up of macromolecules that give sticky character to the cell. It gives what? Sticky character to the cell. Glycocalyx differs in composition and thickness among different bacteria. It is in the form of a loose mucilaginous sheath, and is also called as slime layer or thick and tough covering called the capsule. It is also called as the slime layer and is also called as the capsule. Okay. Now, what is the function of this glycocalyx? The function of this glycocalyx is that it helps in resisting phagocytosis. There is no phagocytosis of the bacteria. What do you mean by here? Phagocytosis. Phagocytosis means killing of bacteria. Phagocytosis here means killing of bacteria. So this mucilaginous sheath, this mucilaginous sheath, this mucilaginous sheath, it protects, it protects, it protects the bacteria from getting killed, from getting killed. Okay. So this was about the mucilaginous sheath. So what is the function of this mucilaginous sheath? The function of this mucilaginous sheath is that it helps in resisting phagocytosis. So this was about the glycocalyx, the outermost layer. This is the outermost layer of the bacteria, which is called as glycocalyx or the mucilaginous sheath or the capsule. Okay. Now the second one is the cell wall. The second cell envelope here. This is the cell wall. This cell wall, as you're seeing, this is the cell wall. Okay, so this cell wall is present just below the glycocalyx. This cell wall is present just below the glycocalyx and it is made up of peptidoglycan. So now we'll study about cell wall. Cell wall of bacteria. Cell wall of bacteria. Okay, cell wall of bacteria. Now this cell wall is made up of peptidoglycan. It is made up of peptidoglycan. Okay, it's made up of peptidoglycan, or we can say murine Okay, and this cell wall is rigid. This cell wall is what? Rigid, and it is solid. It is rigid and solid, and it gives shape and strong structural support to the cell. It gives shape. And strong structural support to the cell. Okay. Now, what is the function of the cell wall? Functions of cell wall. Function of mucilage sheath was that it helps in resisting the phagocytosis. It helps in resisting the killing of bacteria. Okay, this was the function of glycocalyx. Now the cell wall functions. Firstly, the functions of cell wall. It helps in preventing the cell from bursting or collapsing. It helps in preventing the cell from bursting and collapsing. Okay. Then second, it allows the material to pass in and out of the cell. It allows materials to pass in and out of the cell. Okay. Then C, it wards off the track of the pathogens like viruses, bacteria, fungi. It does not allow the attack. It does not allow the attack of pathogens. What are pathogens? Pathogens are disease-causing microorganisms, which may be bacteria, virus, fungi, okay, or protozoan. Now, the last function of the cell wall is it provides mechanical support to the cell. Provides mechanical support to the cell. It provides mechanical support to the cell. Okay, so these are all the functions of the cell wall. Now, the third one, the third innermost membrane of the cell envelope is the plasma membrane. It is what? The plasma membrane. Okay, now this is the innermost layer. Innermost layer. This is the innermost layer of cell envelope. It is the innermost layer of the cell envelope. Okay, it is semi-permeable. Plasma membrane is what? Semi-permeable. In the this I am telling you about in the case of bacteria. This semi-permeable. What do you mean by here semi-permeable? Semi-permeable means that it allows the transfer of certain particles to pass through it. It allows only certain particles to pass through it. So it is semi-permeable and is responsible for the interaction of the cell with the outside environment. Okay. Now what is the function? What is the function here? The function of the plasma membrane. It helps first. It helps in regulation of the exchange of specific materials. It helps in the regulation of the exchange of specific materials okay. Then between the cytoplasm and the extracellular medium. Then second function selectively permits particular molecules to pass and prevent. It selectively permits certain molecules to pass through it. Certain molecules to pass through it. Okay. Then the next function is it prevents the loss of components of the cell. It prevents leakage. Okay. Then fourth one is the plasma membrane is vital to the cellular homeostasis and therefore the health and welfare of all living organisms. So this plasma membrane maintains the internal environment of the cell. Maintains the internal environment of the cell. Okay. So this was about the plasma membrane. Now the next topic which we will study about is the genetic material. The genetic material in a bacterial cell. Okay. Now, as we all know, the bacterial cell is what? A prokaryotic cell. This is a prokaryotic cell. Okay. So, nucleus represents the genetic material of the cell. Okay. Nuclear, it means the genetic material of the cell. Okay. And this is a prokaryotic cell and the nucleoid is naked. The nucleoid is naked. Naked means it is not bounded. It is not bounded by a nuclear membrane. It is not bounded by a nuclear membrane. Okay, it is not bounded by the nuclear membrane. Many bacteria, they contain a small circular DNA As I have told you, this is the bacterial cell I am drawing here. This is the cell envelope. Okay, this is the cell envelope. Here the outside, it is what? The capsule. This is the capsule. This is the here cell wall. This is the plasma membrane. This is the plasma membrane. Plasma membrane. And here you have the nucleic acid here. Okay, this is the nucleic acid. This is the nucleic acid. This is prokaryotic. Nucleic acid is without any nuclear membrane. Here inside there is what? Cytoplasm. This is the cytoplasm. Okay, and there is no doubly membrane bounded organelle in the cytoplasm. Since it's a prokaryotic cell, you will find ribosomes. You will find what? You will find ribosome. These are the ribosomes. Ribosomes. And no doubly membrane bounded organelles, like there is no mitochondria, no endoplasmic reticulum. Okay, no Golgi apparatus, only ribosomes can be found here in the cytoplasm. So this nucleic acid is without any nuclear membrane, therefore it is called as nucleoid. Okay, now many bacteria, they contain a small circular DNA. Many of the bacteria, they also contain a small circular DNA like this. This small circular DNA is called as plasmid. This small circular DNA is called as the plasmid. Okay, this small circular DNA is called as the plasmid, other than this genomic DNA. Okay, these plasmids, they transfer certain unique characters to the bacteria. They have certain genes. Okay, these plasmid, they have these plasmid or these circular DNA, they have certain genes which confer certain unique characteristics to the bacteria, which gives certain important characteristic to the whole bacteria. That is that can be antibiotic resistance. So there are certain genes which is found in this plasmid, which gives certain characteristic properties to the bacteria, like the antibiotic resistance. Okay, so this was about the general structure of the bacteria. Okay, as you are seeing in this diagram, I have told you this is the cell envelope. Okay, this is the nuclear body, nucleoid without any nuclear membrane. There are the ribosomes. They are polyribosome. What do you mean by polyribosome? When ribosomes are attached to each other, okay, and they form a chain, then it is called as polyribosome. Here this is the outer sheath, mucilage is there, plasma cell wall is there, plasma membrane is there. Then this is the cytoplasm. Okay, this is what the flagella. Here you can find here the flagella like this. This flagella, it is the locomotory organ. This is the flagella which helps in the locomotion of the bacteria. Okay, so then the bacteria is also covered with certain fimbriae. This fimbriae helps in attachment of the bacteria to the rough surfaces. Okay, then pili is also there. Pili helps in the sexual reproduction of the bacteria. So this was about the general structure of the bacteria. So this much the lecture is over now. Please, if you like my channel, please subscribe and like my channel. If you find this video lecture as informative, please subscribe and like my channel. I am Dr. Nidhi Sharma with my bio classes. Okay, thank you.